to come in through. Hi, Alex.
Hi, Alex. Hi. Sorry about this. It's just there's some hardware issues. I don't know if you can hear me.
Your voice a bit
okay.
bassy. Oh, okay, that's better.
Okay, I apologize about it.
Okay, perfect. So I think we can start. So it is a real pleasure to introduce Tumi today, who's going to give her thesis talk, and Alex will say a few words about Tumi. For me, it is to start, it is to introduce the committees. I'm the chair of Tumi's thesis committee. Alex Kolakov is her mentor. We have Lee Moore, Joshua Tor, Justin Keeney for Cold Spring Harbor, and as an external, we have Dima Rinberg on today's call. So Alex, if you want to say a few words about Tumi.
Yeah, it's my pleasure to welcome everyone, and thanks to everyone for logging into this. It's my pleasure to introduce Tumi. Tumi came to my group without much programming expertise, and so the result of her work mostly relied on dealing with modern AI packages, artificial intelligence packages. So you can imagine how much she had to learn, and I should say that she did it very well, and she essentially built the entire framework that she will be describing from the ground up. So I'm really proud of this achievement, and it was great to see her grow like over the last few years. I think like everybody wants to get their PhD, of course, but I think with this PhD there will be this dubious honor of being able to be called Dr. Sniff. So there will be at least 2 people now who will be called Dr. Sniff. So without further ado, it's my pleasure to introduce Tumi and her thesis.
Hi everyone. Welcome to my talk of pre-Dr. Sniff. So today I will tell you my work at the intersection between neuroscience, chemistry, and machine learning, titled Deep Nose, using neural networks to represent the space of tolerance. So just an overview of what happens in your brain when you smell something. You have a cup of coffee, there are odorant molecules that they are volatile, and they are carried through air into the olfactory epithelium in the nasal cavity in this box here. There in the nasal cavity, they interact with sensory neurons. These neurons express receptors on the cell membrane. If we zoom in here, and these receptors in these green and blue colors, they are called olfactory receptors. When odorants bind to these receptors, the neurons are activated. So thus the olfactory receptor convert a chemical signal to an electrical signal in the brain. You may notice that there are many different colors here, and that's to illustrate the fact that there are many such olfactory receptor genes in our genome. Each neuron expresses a single type of olfactory receptor genes. In addition, as you may see, while the neurons intermingle at the epithelium, their axon project to 1 or 2 specific sites in the olfactory bulb, and these sites are called the glomeruli. So just to sum up, as an example, these green neurons express green receptors, and axons of many different green cells converge to the same glomerulus. As a result of this wiring pattern, each glomerulus now represents one type of receptor. And when you image the activation pattern of these glomeruli, for example, in animals, you obtain a neural pattern that encodes for the odorant identity and concentration. This neural activity is then subsequently carried to other regions of the brain, such as the olfactory cortex, to allow perception of odorants. So that's why we say that these olfactory receptors are involved in both the detection of odorants with this binding of odorants at the membrane, but also as involved in the representation of odorants with this glomerulus map here. So during my time in the Kulikov's lab, I'm involved in a few projects. The work that I'm presenting today is the first one that relates to the detection and representation of odorants. In addition to this project, I also work on theoretical models for the organization of the olfactory bulb, as well as contribute to a reinforcement learning project led by Sergey in collaboration with the Bolis lab. So in the interest of time, I will focus on the first work. So just to repeat in a slightly different manner, what I have shown you in the first slide, we can frame the problem of olfaction in terms of the processing of information, where you have an odorant stimulus that interact with an ensemble of olfactory receptors. These receptors then activate neurons, and these neuron activity reflect the identity and concentration of the odorant stimulus. Further, we have processing of the olfactory bulb and the cortex that gives rise to the perception of odorants, such as this smell like fruity or floral, etc. Because this neural activity encodes property of odorants, for the rest of this talk, I'm gonna refer to this property as the chemical space. So this problem of representing the stimulus into a sensory space is not unique to the sense of smell. For example, in there's some typing. For example, in color vision, we have 3 photoreceptor genes, and these 3 receptors are tuned to different range of wavelengths. And we know what these range are on the right here. On the other hand, olfaction is more complicated. First, we have a bigger numbers of receptors. So we have 350 GPCR olfactory receptor genes in human and more than 1,000 in mice. Second, it is not well understood, although some experimental work has been done, what kind of sensory features each of these olfactory receptors gene is responsive to. And as a result, we don't have the same understanding of the sensory space of olfaction compared to vision. So that inspired my work. So in this work, I aim to study the chemical space of odorant molecules and the factors that influence the composition of the receptor ensemble using modern machine learning framework. So here, we use neural networks to model the olfactory receptor ensemble. And this network will convert an odorant input to some output, and the output of this network is analogous to responses of olfactory receptors. I will explain about neural networks in subsequent slides. And just to put this work in the context of the field, there are many ways to represent the chemical space using computational models. Our models differ in 2 ways. First, we differ in how we represent an odorant input. And the second, we differ in how we obtain a chemical space. So what do I mean by that? So in terms of the odorant input, you have to describe this entity of chemical to the network in some form. And we choose to describe these molecules as 5-dimensional objects. So if you look at this example on the slide, we have chlorobenzene. This molecule has 6 carbons, 5 hydrogen, and 1 chloride. We can represent each atom using a 3-dimensional Gaussian cloud, and we can put this atom into different channels according to the type of the atom. So for example, the carbon the 6 carbon atom can be in the carbon channel, and the hydrogen atoms into the hydrogen channel, etc. So this is analogous to when you have a two-dimensional image and you have different channel for the colors. So here we have different channel for the elements. So far I have covered 3-dimensional of space and the 4th dimensional of element. And the last dimension is the orientation or conformation of molecules. When you look at 3-dimensional objects, they can take up different configuration in space. And so we include those different configurations in the 5th dimension. So to sum up, we have 5 dimensions. And why did we decide to include 3-dimensional structure? There are many ways to describe a molecule, such as using a graph where each atom is a node and a bond is an edge. We choose this representation because we know that the node is chiral. And what that means is that if you look at this example, these 2 molecules, they are mirror images, but they smell different. So these are R carbon and S carbon. If you use the graph representation, these 2 molecules would be identical unless you manually specify each chiral center. But with 3-dimensional representation, you can have enough information to discriminate between the 2. So that's for the input. And as for how do we obtain chemical space, one way, and the way that we choose, is to discover these chemical features that are relevant to olfactory perception automatically from data. And what this means is we use odorants data set, which I would describe in the later part of this talk, and train a network to produce a certain output. And the features that was learned by the networks after training would constitute this chemical space. Alternatively, other methods, instead of using data and learn automatically from data, we can use human knowledge. So this is called feature engineering as opposed to feature learning, where a chemist can look at an odorant molecule and describe it in terms of molecular weight, atom count, ring count, but also there are many other complex properties that can be calculated. And these 2 spaces may be similar. Maybe it's enough to describe using human knowledge, and that would be the similar features that the olfactory system care about. But our method, we do not make any assumption. We obtain the space from data, and we will compare these learned features with human engineered features. So to sum up, what we do in this work is we describe odorants input using a 3-dimensional structure, and we train the neural networks to learn the chemical space automatically from data. So far I have mentioned a lot about data. So what kind of data do we have? We have a group of molecules on the order of 1,000 that we know how they smell. So I'm giving 2 examples here. This molecule on the left smell floral, fruity, jasmine sweet. So these are called semantic descriptors, where you use a word to describe a molecule, and each molecule can be associated with 1 to multiple words. Besides these molecules called label molecules, we have a much larger number of molecules with no order annotation. So we have on the order of millions of molecules where we know the structure, but we don't necessarily have a human perfumist to test this and annotate them. So in this kind of work, we leverage both types of data. We use both label molecules and unlabeled molecules. We do this because we are inspired by machine learning research in other modalities such as language and image, where you have a similar issue. You have a great abundance of unlabeled data sets, such as this image you can see on the internet, but you have fewer labeled images. So the strategies used by those studies and also in our study is to first perform pre-training to learn the chemical features from unlabeled data set, and subsequently to fine-tune on labeled data set. So to sum up, this led us to implement 2 stages of training. In the first stage, we perform unsupervised learning with unlabeled data, where we train the network to learn to reduce dimensionality of this large library of millions of molecules. And in the second stage, we would perform supervised learning with labeled data, where we would use molecules with annotation and train a classifier to predict those annotations. So to dive into the first part, we first train an autoencoder. So this is what this architecture called. This autoencoder consists of an encoder which transforms the molecule input, which was 5-dimensional object that I mentioned previously, into a latent representation with about 50-fold fewer units. And from this compression, the decoder needs to reconstruct the input. And so we train this network on the library of unlabeled molecules. So you can see that the deep nodes, the middle layer, the compressed representation, we call it the deep nodes features because this is the deep nodes network. It learns deep nodes features. These features contain information about identity of odorants, and therefore we think they are analogous to responses of olfactory receptors to odorant stimuli. So these networks, you could see I keep drawing these circles and arrows. These networks, they are created by having multiple layers, which is the vertical stack of circles here, stacked on top of each other. Each layer would convert information from previous layer to the next, and we use the arrows, the type of filters we use. And next I'm going to explain a specific type of layer we use called convolutional filter. In case you're not familiar with convolutional filters, you can think of them as feature detectors. So just look at an example of this picture here. So you have an image. On the top is the pixel intensity of this image. And on the bottom is what the image look like. So you can see this simple image. It has, on the left, it has a patch of light color pixel associated with pixel intensity of 1. And on the right, it has a patch of dark color pixel with intensity 0. And the filter is in the middle. So this filter is called a vertical edge detector. And let me show you real quick how it achieved that. So if you apply this filter to small patches of the image, so let's first apply it to the first patch of the image. What you do is you just multiply individual component of the filter and then sum up. So you can see that the +1 and the -1 after multiplication, they cancel out. So you have an output pixel intensity of 0 after applying this filter to this patch of the input image. And you can repeat this operation for over the entire image. So in the second patch, you also have this cancel out of the +1 and -1, resulting pixel intensity of 0. But here at the area where you see a change from light to dark color, the +1 is no longer canceled out by the -1 because it's 0 here. And so as a result, you have a pixel intensity of 2. So if you just keep doing this for the entire image, you would get an output image with pixel intensity right where the edge is. So that's why we call these kind of filters edge detector, but you can also have various filters for different kind of features. So this filter is an example of 2-dimensional convolutional filter. In our work, deep knows because molecules are three-dimensional objects, we use three-dimensional convolutional filters. And we do not predetermine the value of the filters like in this case, but instead we would learn it. And what do I mean by that? So again, the arrows are the filters that I just talked about. For filters, we would first initialize these values with random numbers. And as a result, the initial reconstruction would not be accurate, as you can imagine. But as we provide a network with more examples to learn on, we can iteratively update the value of these filters to reduce the error. So here's an example of that process. So as you can see, the top row, so this is an example of a molecule in the test set where and this means the network has not seen this molecule before. On top is the input molecule and the bottom is the reconstruction. So as you can see, when we initialize the value of the filter randomly, the reconstruction is very inaccurate. And I'm gonna play the video, and this shows that over time, the network learned to better reconstruct by modifying, adjusting the weight of these filters. So you can see the accurate reconstruction in every channel. So each of these channels are carbon, hydrogen, oxygen, etc. Here's some more examples on the left. You have, so each of these boxes are the two-dimensional projection, but each box is actually three-dimensional. So you have the input and output after training the network. So these are just some example molecules, but this is also true if you just look at the entire test set. You can see that on the y-axis is the correlation coefficient between the input and output pixel. And you can see that the network reconstructed accurately in each element. So far we have shown that we were able to train this autoencoder network to accurately reconstruct the molecules. And I mentioned before, the deep knows features are the learned representation of odorants, and we would like to compare it with the human engineer counterparts. So how do we do that? This table show on the left column are the first three principal components of the deep knows features, and the right columns are the human engineer features that are most correlated with these dimensions. So principal components is when we reduce dimensionality of the data set and we obtain the direction with the greatest variability in the data. And so we see that the first principal component correlates with molecular weight. The second principal component correlates with the number of bonds to oxygen, and the third principal component correlates with the number of rings. So just to show in a more visual way, what I did is I looked into this space. The first three, so this slide shows the first principal component, and I randomly sample molecules in small bins along this direction. So we go from small value of this principal component to large value. And as you can see, it's just very clear, even visually, that you go from larger molecules to smaller molecules. So this principal component negatively correlates with molecular weight. The next figure is the third principal components, and it correlates with the number of rings. So we go from these molecules having ring to more, like rotatable, more straight kind of molecules. So just to sum up, we for the first part, we train an autoencoder to reconstruct the molecules after substantial compression, and we found that the learned features correlate with human engineer properties such as molecular weight and the number of rings. So since we think that these deep nose features are analogous of responses of olfactory receptors, the next thing to validate these features is also to use it to predict human olfactory percepts. And to do that, let me introduce the data set that we use. So we have human olfactory data set, and I briefly mentioned it before, where we have molecules paired with some labels. So these are some example of molecules with the semantic descriptors. And there are many of these data set, and one of the data set is the Leffingwell data set that I'm going to talk about today. This data set has more than 3,000 odorants. So these molecules here, and more than 100 descriptors, so about 113. So descriptors are these words such as fruity, meaty. And so as you can see, this response matrix would be binary. So a molecule is either fruity or not. So that's the kind of data set that we're dealing with. So since it's binary, we're training classifier. And the way we do that is we start with the Tran autoencoder. And you have seen this figure previously. But our goal is not to reconstruct the molecules, but to predict the perceptual quality of the odorants. So to do that, we would replace the decoder network with a classifier network. And we would do the same thing. We would modify these ways to match the prediction. And so there are two things we're interested in learning from this kind of experiments. First, we would like to know that if the features learned by the autoencoders can generalize to prediction of olfactory percepts. And second, we want to know if there are actual advantage to learning features from data compared to human engineered features. So let's answer that. So I'm going to explain how we answer the first question. So we would like to know if the features learned by the autoencoder, which was to reconstruct this molecule, can they generalize to prediction of olfactory percepts? So recall that the weights of the network are iteratively updated to produce an accurate output. In one of the network and on the top, after training the autoencoder, we would fix the weight of the encoder. We no longer modify it. We only modify the classifier. We only train the classifier. So this is to test if the encoder features can generalize. But on the other hand, we also train a network where we train the weights of both the encoder and the classifier. So we would compare these two to answer that question. And the second thing we're interested in is whether the learned features are comparable to the human engineered features. And to do that, we also train two different networks. One network, as I have talked about before, where we have the encoder and the classifier. And the second network, instead of using the encoder to generate these chemical features, we would use software. And the software we use is the moderate software. So this would help us compare the two types of features, learn versus human engineer. So to sum up, we have three features that I'm gonna compare: the human engineer features, the pre-trained features learned from the autoencoder versus the fine-tuned features. On the x-axis is AUROC. So these are performance of the classifier. A value closer to 1 means that the value produced by the classifier can perfectly separate positive and negative examples. And a value closer to 0.5 means that they cannot separate the positive and negative example. So we would like something closer to 1. So what we could see from this plot is that, first of all, if you look at the first two rows, you can see that the pre-trained features can predict some aspect, but overall is not as good as the fine-tuned features or the moderate features. And the second point is that the fine-tuned features are more predictive of olfactory percepts compared to moderate features, suggesting that there's benefit to learning chemical features from three-dimensional representation. And I showed you previously that there are over 100 descriptors. So I'm just showing a histogram of how just an average predictions score of this predictor. So certain words, what this plot is showing is that certain words are better predicted compared to others. So it's easier to predict, the network perform better to predict in predicting garlic and phenolic compared to features such as warm and creamy. So we have obtained, so what I have shown you is that we fine-tune the network further, and we obtain an updated set of deep nose features that is more predictive of olfactory percepts. So we would like to analyze this chemical space because we think it's relevant. It encodes for chemical features that are relevant to olfactory percepts. So there are a few things that we can analyze when we look at these features. And I'm gonna go through this one by one. So first, we're interested in the structure of the learned chemical space. And one thing you could do to just have an idea of the structure of this space is, again, using principal component analysis. Take the two dimensions that are most prominent and project this data onto this dimension. So here, if you just look at the first panel, the x-axis is the first principal component and the y-axis is the second principal component. Every dot is an odorant. And dots that are colored orange are odorants with vanilla descriptor. And everything else doesn't have vanilla. And these 4 different panels, I color 4 different descriptors. So what you can see here is that we see that in this space, we see that the odorants that share the same descriptors are located near one another. So there's a local structure. But you also see a global structure where you see that odorants having different descriptors occupy different regions in this space. And more than that, you have regions that are overlapping when you have descriptors that are more similar perceptually to another. So for example, pineapple and pear are both fruity descriptors, and you do see that molecules with these 2 different words, they are in nearby regions, at least in the first 2 principal components. So the next analysis that we can look at is to look at the dimensionality of this space. So to do that, again, we did a principal component analysis, but we look at the explained variance, the cumulative explained variance on the y-axis. So just follow one curve, for example. This green curve, which is the pre-trained features obtained after training the autoencoder. The way you interpret this plot is that using 10 principal components, you can explain about more than 90% of variance in the data set. So you can estimate that the chemical space of these features are about 10 dimensions. Whereas when also look at 10 principal components for the other 2 features, the human engineer and the fine-tuned features, it explains less variance in the data set. So these two spaces have higher dimensionality. So this could explain why the pre-trained features doesn't perform as well as the fine-tuned features or the human engineer features. The next thing we looked at is how this chemical space changed after fine-tuning to perceptual prediction task. And to do that, what I did is, again, I previously showed you where we calculate the correlation between the deep nose features, principal components with the human engineer features. And previously, I showed you that molecular weights, for example, correlates with the first principal component. But then what I do now is I look at how this correlation changed after fine-tuning to perceptual prediction task. And what we see is that the chemical space of 10 after fine-tuning to this task, they look different. So for example, these features care more about number of sulfur, the number of aromatic ring, the topological surface area, than before when we just train the autoencoder to reconstruct the molecules. And on the other hand, features such as molecular weight are still important, but to a lesser degree. So to sum up the three type of analysis that I did on the deep nose chemical space, we first found that this space reflects both the local and global perceptual relationship. Second, we found that the dimensionality increased after fine-tuning with the supervised learning. And last, we saw that the network prioritized different chemical features after fine-tuning. So lastly, I would like to show some kind of visualization, because so far I've shown, a lot of numbers, these performance numbers, but it would be nice to see some kind of visual analysis. Next, I would show you a type of analysis that can be done to calculate the contribution of individual atoms to prediction of perceptual descriptor. So if you look at the top row, I'm showing two molecules that are close together in the chemical space. So what it means is these molecules, they look similar, but they have very different percepts. So on the top row, I show one pair, and the second row, I show the other pair, where one molecule is sweet and honey, and the other is meaty, roasted, and sulfurous, with only two change in the structure. You have a nitrogen here instead of a carbon, and here you have a side chain. So how do we do that analysis? Again, we learned from in the vision modality, where people have trained neural networks to classify objects in an image. And this analysis is published, and it's called occlusion analysis. What people did is, if you look at the first image, the classification of this image is Pomeranian. And what you can do is you can see what pixel of the image contribute most to this prediction by occluding individual patch in this image and classify this network. So if this patch of pixel is important for the network to identify the dog, you would see that the classification score decrease. So this network, after you do this analysis on the first picture, you see that it highlights this region near the face of the dog. Similarly, the second picture, you have classification of the wheel, and you do see indeed that this patches image of the wheel affects the prediction. So we do the same thing for molecules, where I'm just going to come back to the previous example. And what we would do is we would occlude the pixel associated with individual atom and redo the classification. So I'm just gonna overlay this picture with the molecules where I color the atom contribution. So just to guide you, so red means that this atom contribute positively to the prediction of the descriptor. And just focus on the first row. There are many descriptors here, but this analysis is done on the fruity percept. And we know that molecules with fruity tend to have an ester group. So you do see indeed that this ester atom, the atom in the ester group have the red color, so they contribute positively to prediction of fruity. On the other hand, in this molecule that doesn't have fruity, this sulfur, the presence of this sulfur really decrease the score of fruitiness. So this visualization can help you see that kind of atom level contribution. And the second row is when you have the percept sweet on the left, and the right doesn't have the sweet annotation. And you could see that it's the presence of nitrogen here that really decrease the prediction score. I'm just going to show a few more examples, and these examples, they align with I think what we know about the garlic smell. So sulfur contribute to the smell of garlic. So you can see in these two different molecules, the sulfur color red after this analysis. And the smell phenolic, here are two different molecules with phenolic smell. And you do see that atom associated with benzene ring contribute to the prediction of this descriptor. The next is spicy smell. So it's really important to have this chain, this side chain with methoxy group. They contribute to spicy prediction. In terms of floral, you have small contribution from many atoms. But some of the other descriptors, such as nutty, you really see this like intense contribution from these two nitrogen atom on the ring. So far I have shown you one descriptors and different molecules that's annotated with those descriptors. In the next example, I'm showing identical molecule, but the difference is the different color. So on the left, I color by its contribution to the sweet percept of the same molecule, and on the right, I color according to the contribution of the atom to phenolic descriptor. So you can see that different atom, when a molecule have different notes of smell, sometimes different part of the atom contribute to different percepts. So for example, for the sweet, it's more this 2-methoxy side chain, whereas for phenolic, it's really the atom near the ring, in the benzene ring. So just to summarize this talk, I first talk about how I train the neural networks to represent the space of odorants. This chemical space is learned from data. It used 3-dimensional molecular structure as input. We further validate that this space is predictive of perceptual property. We found that after fine-tuning to predict, to classify perceptual descriptor, this space is tuned to different kind of physical chemical properties, suggesting that these are features that the olfactory system cares about. And lastly, we perform occlusion analysis to visualize atom level contribution to perceptual prediction. So the next slide is to just tie it up, because this work it's a machine learning work, but it's inspired by neuroscience. So how can so how these two fields can benefit from one another? So in this work, It's just like two figures to kind of like talk about the parallel between the biological system and the neural network system. You have this encoder that try to model the olfactory receptors, the responses, the deep nose features is analogous to the responses of neurons. And the classifier is trying to produce a perception of an odor. So work in many labs, for example, Florin Albienu here right at Cold Spring Harbor is recording in collaboration with the Kulakup's lab. He recorded glomeruli activation. So basically, we will soon have access to the real neural activity in the glomeruli along with identity of the receptor. And once we have that information for sufficient number of molecules, we can use this to guide the design of the network. So right now, the network's only objective is to produce correct classification. But if we have further information about neural activity, we can update we can add another criteria, is that it not only have to provide correct classification, but also needs to imitate some kind of statistics of the real biological system. So that future work. And just also various aspects that this work hasn't captured. So far, I have shown you how I predict a single odorant. But in reality, this receptor responds to mixture of odorants, and sometimes the effect when you have a mixture is not simple, as it's not additive. Sometimes having one mixture component antagonize receptors, so that would be for future development. And right now, I only include the atom identity. But we are also working on including the bond type. So right now the network only has, this oxygen, hydrogen, and carbon, but we could certainly include other information such as the single bond, double bond, etc. And lastly, just to point out that the network that we use in the classifier is a single layer feed-forward network. But this is not what we know is the case. For the connectivity in the olfactory bulb and the cortex, we have recurrent connectivity there. So that's what this work has yet to capture. And I want to end this talk with acknowledgement. I would like to thank the Kulakup's lab, obviously, but also my thesis committee. Pavel Olsen is the chair, and this work of occlusion was inspired from a discussion in the thesis committee where Pavel coined the term shape of pleasantness. So I was very proud of that term. It's very catchy. Limor is on my committee, but she's also my academic advisor. And I admit that I admire her work even before I come to Cold Spring Harbor. So she gave me many useful feedback, both as my committee member and as my academic mentor. And Justin gave me very useful feedback on my thesis, but also organized many QB activities. That was great. And my examiner is Dima, and one of the most inspiring day I had as a grad student was the day when Dima invited me to visit his lab in NYU to present my work, but also talk to many grad students. So that was really fun. And I want to thank everyone at the staff at the School of Biological Science for making this PhD as free of bureaucracy as possible. And like, since I'm doing this very, computing heavy work, and as Alex mentioned to you before, I didn't come with a programming background. I got a lot of help from Todd and Osama on computing related matter. And it was great. Like sometimes I phrase the question very awkwardly, but they always managed to understand what I meant to say. And other campus resources such as the library, the Writing Resource Center that helped me with various manuscripts, And also, it's very important that at lab we have many professional development activity. I did this walking tours, and it really helped me improve my science communication skill, and also the Bioscience Enterprise Club and Women in Science and Engineering provide great professional development activity. And since I just thought it would be on brand for me to present an extra data slide where I have other people I would like to thank. But as you can imagine, acknowledgement is a high dimensional space. So what I did is I performed dimensionality reduction on the space of people I would like to thank. And so I color some people here. And you can see pattern in some of the cluster. So these are the first two dimensions, and you can see this cluster is the lab mate. So you know, it's this has been a fun experience, and we not only get along our scientific partner, but we also get along well outside the lab and enjoy various activities together. And then you have this cluster that are friends and classmates, and it's a great cohort, and I have learned so much from everyone. It's also like this picture was taken during the pandemic, and I was very grateful to have this friend bubble to help me get through this pandemic. And I learned that cats are cute, and I improved my running muscles as well as my drinking muscle. This next cluster here is my current advisor, Alex Kulikov, and also my former advisors at Novartis and also at San Jose State. This is my chance to embarrass Alex. I'm just gonna like zoom in to this gorgeous picture of him. We were at a Yandex party at ICML conference, and this cluster is the group of friends I made in the post-bac program at Novartis. This program prepared me for grad school, but also let me be part of this group of people, and many of them joined grad school the same time that I did, and we still keep in touch. So it was very encouraging to just have friends in other places going through the same journey. And lastly, this is my family. So not so much in like the professional cluster here, but they really gave me, great upbringing, including good food and good education. So my mom's not here today, so that's why we're doing the recording. And so then, there's future direction, and there's some data point that I don't yet know how to put in this space. So we have a new family members, my niece. And from this picture here, you can see, obviously, she could make a good coworker. She's very engaging. I can make slides with her. We discuss a lot, although I don't know what she's talking about a lot of the time, but she did teach me about both pleasant and unpleasant odors. So I could put her here in the lab mates or here in the family. So that would be for future direction. And now I welcome a question from the audience, and maybe we can turn off the recording so people are comfortable asking.